Hi everyone, welcome to my YouTube channel Biology by Kelly said. Today I will be discuss about molecular basis of inheritance. First of all, uh, I will discuss about introduction of genetics. You know that genetics is the science of biological inheritance, that is the causes of resemblances and a difference among related individuals. That means all types of parental characters, how to transfer into the daughter generation. Genetics may also be defined as the science that deals with the nature and behavior of genes, the fundamental hereditary units. That means genes are the basic unit of inheritance that are responsible for behavior of living organism and his nature of living organism. Basically genes are related to the all types of metabolic activity, vital activities which are takes place into the living organism. The term heredity indicates like begets like, that is offsprings tend to resemble their parents. For example, you seen in any types of living organism, a parent and a daughter generation are very similar to each other. Genetics occupies a central position in modern biology. It can be applied to all microbes, plants, and animals also. Into the next slide, I will discuss about branches of genetics. Basically, genetics are divided into three main branches. First one is the transmission genetics. What is the transmission genetics? It is also known as classical or Mendelian genetics. Is the oldest branch of dealing with the transmission of genes from generation to generation. Second one is the molecular genetics. Uh, Molecular genetics deals with the structure and function of genes at the molecular level. And third one is the population genetics. Population genetics deals with the distribution and behavior of gene, usually in mathematical terms, in groups of interbreeding or related organisms called population genetics. These are the basic branches of genetics. In this slide, I will discuss about genetics terminology. There are some genetics terminology which are very useful in genetics. First one is the trait, an inherited character that are detectable variant, and that means those characters which are transferred from parent into the daughter generation and responsible for the variation into the daughter generation. Second one is the cross, a mating between two individuals leading to the fusion of gamete. That is male and female gamete fusion. You know that formation of male gamete is known as male gametogenesis and formation of female gamete is known as female gametogenesis respectively. You know that the formation of gamete basically takes place in the reproductive system of organism, and the result of fusion of gametes is known as fertilization. After fertilization, a new individual are formed which are totally similar to his parents, and sometimes uh, few amount of differences are seen into the uh, daughter generation. Unit factor, that is gene, the determinant of the characteristics of organism. That is what is the reason, what is the factor which are responsible for the transfer of inherited character from parent to daughter generation. Factor is also known as gene. The locus or gene locus. What is gene locus? The specific place of a chromosome where gene is located here. That means you know that basically the genetic character, that is the nucleic acid DNA is also contain gene, and nucleus basically and DNA are condensed to each other to form chromosome. And chromosome That place of chromosome that are contain genes is known as locus. Next one is the alleles. Alleles, alternative form of a gene, for example, capital T and small t alleles, represents tall and short stem height of pea plant. You know that basically the different characters which are used by seven opposite character in Mendelian inheritance are also known as alleles. Next one is the homozygote. What is homozygote? Homozygote and individual having two identical alleles, for example capital T or small t. These are the pure characters of parental generation. Next one is the heterozygote. Heterozygote and individuals having two different alleles. For example, capital T and small t. Capital T and small t are also responsible for the hybrid character into the F1 generation. These are result of cross between capital T and small t. Uh, Next one is the phenotype. Observable morphological appearance of the trait, for example, tall plant or red flower. That means morphological character of any living organism is known as phenotype. Next one is the genotype. What is genotype? Genotype is the genetic constitution of an individual. For example, capital T and small t is also known as genotype. So what is difference between phenotype and genotype? Phenotype is morphological condition or morphological mm, character, but genotype basically related to phenotype, that is the gene or responsible for the phenotypic character. Next one is the hybrid. Offsprings obtained from a cross between two genetically different parents. That means pure plant and dwarf plant are crossed to each other. The result of into the F1 generation obtained a hybrid plant. That hybrid plant are also known as hybrid nature of organism. These hybridization are basically related to the pureness and dwarfness. What is the conditions for calling hybrid? That is the some basic principle of a hybrid. That means only dominant character are seen into the F1 generation, but recessive character are not seen into the F1 generation. And this is the main reason of called hybrid. P1 generation, sorry, P generation, the organism involved in the original cross. That means parental generation is also known as P generation. Next one is the F1 generation. What is F1 generation? F1 generation is also known as first filial generation. That means generation of hybrids obtained from a cross between two genetically different parents. Second one is the F2 generation. It is also known as second filial generation. That means individual obtained from the breeding of hybrids of F1. Next one is the pure line, a series of self-breeding generation having homozygous genotype. That means a result of self-pollinated pea plant and self-pollinated organism is known as pure line. Next one is the phenocopy, non-hereditary phenotypic modification caused by special environmental condition. Some conditions, you know that any types of living organism which are totally similar to the parent and daughter generation, because there are no difference between parents and daughter due to environmental issues. I will discuss about some landmarks into genetics. Uh, there are some scientists which are play most important role in the field of genetics. Uh, in 1886, Gregor John Mendel gives basic principle of heredity. In 1871, Frederick Misser isolated nuclein from the nucleus. Nuclein, uh, it is also known as nucleic acid, and basically nucleic acid divided into two parts. First one is the DNA and second one is the RNA. In 1882, Strasburger showed that nuclei contain chromosome. Strasburger basically clear that nuclei contains chromosome, and these are related to the genetic material. In 1902, Archibald Garrett identified the first human genetic disease first of all. In 1905, William Bateson called the science of heredity genetics. In 1909, W. Johnson introduced the term gene, which are also known as factor in uh, in terms of in place of gene. In 1953, James Watson and Francis Crick proposed double helical model of DNA. DNA basically five types, you know that A DNA, B DNA, C DNA, D DNA, and Z DNA also. Basically, in books are generally you seen that type of figure of DNA, which are B DNA. In 1973, Herb Boyer and Snelle Cohen first to use a plasmid to clone DNA, that means duplicate copy of DNA. I will discuss about some scientists which are related to the experiment of genetics. So first of all, I will discuss about pre-Mendelian hybridization experiment. You know that there are many scientists are work in the field of genetics to know about what is the reason of inheritance. Uh, Gregor John Mendel was the not first person to have conducted hybridization experiments on plants. J. Coren Uter, which lifespan is 1733 to 1806, a German botanist, had conducted hybridization experiment in the tobacco plant. Corriuter reported that the progeny may resemble any one of the parents or may be intermediate between them. According to Corriuter, the hybrids are more diverse than the parents. Other who had worked on hybridization before Mendel. There are some other scientists include Bartner and some other scientists, for example, Nodin and Gauss also. But these scientists are not clear about what is the reason of inheritance. I will discuss about Mendel's law of inheritance into the next slide. We will discuss about the Mendel's life and Mendel's work also. The first scientific study leading to formulation of laws of inheritance was carried out by Gregor John Mendel, which are uh, lifespan or 1882 and 1822 to 1884, known as father of genetics. Mendel was a great scientist which conducted hybridization experiment in the garden pea, that is also known as Pygium sativum. Uh, Garden pea are the life span of garden pea is basically near about 3 to 4 months, and all types of morphological characters are easily seen in this plant. Mendels published his work in 1866 uh, in a paper entitled experiment in the plant hybridization, experiments in the plant hybridization. Other scientists did not take their work seriously because the Lamarckism and Darwinism was very popular in those days. Mendel's laws were rediscovered simultaneously by three great scientists, namely Hugo de Vries, belong from Holland, Erich von Sermack, belong from Austria, and Karl Karens, he's belong from Germany. These are three scientists which are rediscovered Mendel's law. Mendel's after rediscovery of Mendel's law, Mendel's paper got republished in the journal Flora, a journal which are republished Mendel's law by the three great scientists. Mendels derived two laws of principle related to the segregation. Segregation is also known as purity of gamete and independent assortment. I will discuss about some reasons for the success of Mendel in his experiments. You know that Mendel's was used seven opposite characters which are easily seen in pea plant or Pygium sativum. First one is the stem height. In this type of character, there are two basic character. First one is the tall and second one is the dwarf. Tall character is dominant character and dwarf character is recessive character. And second character is take here flower color. Flower color basically used violet or red, that is dominant character, and second one is the white character. These seven types of characters are clearly seen in the pea plant also. I will discuss about monohybrid cross in this slide, which are very important for clearly understood by the genetics of genetics and Mendel's law. You know that Mendel's crossed parent first and parent second, uh, that is pure tall and pure dwarf plant, and he obtained F1 generation into F1 generation. Basically, all plants are hybrid tall. That means uh, when he will cross with homozygous pure tall and homozygous pure dwarf plant, they will obtained into F1 generation. All plants are hybrid tall because they are heterozygous in condition. That means only dominant character are seen into the F1 generation. Basically, these plants are hybrid in nature, but into the F2 generation, I will discuss in this figure. You know that also Punnett square method. These two hybrid plants, first one this and second one is this. These hybrid plants are crossed to each other and obtained into F2 generation. There are four plants basically. First one is the totally pure tall, and last one is the totally pure dwarf plant, and third and fourth sorry second and third one is the basically hybrid plant. In this F2 generation, you know that 25% plant sorry 25% plant are pure tall and 25% plant are pure dark, but 50% plant are pure hybrid in nature. That means it is also known as genotypic and phenotypic ratio. What is the genotypic and phenotypic ratio into the F2 generation? 3 is to 1 is the phenotypic condition into the F2 generation. And 1 is to 2 is to 1 is the sorry 1 is to 2 is to 1 is the genotypic ratio into the F2 generation. I will discuss about backcross and test cross in this slide. You know that these terms are very important for medical point of view. You know that Mendel are also crossed pure tall and pure dwarf plant, and he will get into the F1 generation. All plants are hybrid tall. These hybrid tall plant, when crossed with his parental plant, sorry, parent plant, then he will get these results, and the cross between F1 generation into parental generation is known as backcross. This condition is also known as only backcross, but this condition is also known as backcross and test cross also. So what is difference between backcross and test cross? Backcross, that means hybrid plant are crossed with his parent plant. Those conditions are known as backcross, but hybrid plant and pure dwarf plant, parent plant, are crossed to each other. Only this condition is also known as test cross. That's all. Thanks for patience.